So dear students, welcome to my video lecture, the zoology, brush up zoology class. We are going to take a topic of embryology. The topic is implantation of embryo in mammals. So before we study implantation of embryo in mammals, we know that mammalian egg is very small. The mammalian egg, it is very small, approximately having this much 0.1 mm diameter. Apparently yolkless, and it develops within maternal uterus, and therefore it has to develop within the mother's body. The blastocyst, it implants on the wall of uterus. So implantation means fixation of blastocyst on wall of uterus, right? So after that happens after about a week after the fertilization has taken place. Fertilization takes place in the fallopian tube. But before we directly jump onto this very topic of implantation, we must have a look what is a blastocyst. So blastocyst, that after 4 to 6 days, we know that after fertilization what happens? Zygote is formed that undergoes cleavage, and after cleavage a ball of the cells is formed which is called as morula. Morula is formed, and we have a diagram for morula. This is the morula. Morula reaches from the fallopian tube. Fertilization has taken place. About 4 to 6 days after the fertilization, from fallopian tube, let's say these are the fallopian tubes, it reaches down into the uterus. And once it reaches the uterus, the morula develops a fluid filled cavity. So this is, you can say, the morula. Morula is having a ball of mass which has, when it reaches down, layer of albumin, the outermost layer of the follicle original, that is zona radiata, and the mass of cells, bulb like mulberry. So this is your inner mass of cells, while the outermost layer of these cells is called as subzonal layer, right? So that is how this morula, it reaches the uterus. Now once it reaches the uterus, the next step is it starts developing, it starts enlarging, and hence it develops a fluid filled cavity. So this is your albumin layer, and here is the zona radiata layer. This layer is zona radiata, and inner to this is another layer which I've already said, it is subzonal cells, and there is inner mass of cells. So cavity develops here in between the subzonal cells and the inner mass. This is a fluid filled cavity. It would develop like this. And further, the cavity enlarges. It goes on enlarging. Once the cavity goes on enlarging, and in the next step, these cells, once the cavities, it is now called as, this is no more called as blastocyst, morula, sorry. This is called as blastocyst, having the same structures, zona radiata. This outermost layer is the zona radiata. This is the albumin, and inner to this is another layer which I was calling subzonal, but we will not call it subzonal anymore now. It is called as trophoblast. And here is the inner mass of cells. In between there is a cavity. So this such a structure is now the blastocyst, or other name is blastodermic vesicle. And the side of the blastocyst, to which embryonal knob is attached, these inner mass of cell which gets confined to one side, that is called as embryonal knob. So this side of the blastocyst towards which the embryonal knob is attached, this is called as the embryonic pole of the blastocyst, or let's say the animal pole of the blastocyst, whereas the opposite pole is called as, this pole would be called as embryonic pole. Is that fine? So trophoblast, this is trophoblast, which we were earlier calling as subzonal layer of cells. Now the terms have been changed, the terminology is changed. So these are called in, these are respectively being called as trophoblast and embryonal knob. Now these cells are no more, let's say the trophoblast cells, they do not participate in formation of any of the embryo proper. They will not contribute to that. Rather than that, the trophoblast would be forming the extra embryonic membranes, which we will be studying afterwards. So this is how this is formed, the structure is formed. And what extra embryonic membranes would be formed? Extra embryonic membranes. The extra embryonic membranes are chorion, amnion. It remains external to the embryo, and these layers are formed out of trophoblast. These membranes, they serve a very important function during development, and later on at the time of birth these would be discarded. Now the trophoblast cells which are in contact with the embryonal knob, they are called as cells of Robber. And now I tell you which are the cells of Robber. The cells of Robber would be, in this previous diagram, these cells which belong to the trophoblast layer, and they are in connection with that of the, these are your cells of embryonic knob, and the upper layer is of, and this is your trophoblastic layer. So these cells one blue, just opposite, just sticking with the inner mass embryonal knob. These are called as cells of Robber. Now at this stage, the blastocyst is about 0.28 mm. It has grown, and its cavity may be compared with blastocele, but embryo is not blastula because cells are already differentiated into different sites. So that was about blastula, and in the next lecture we'll study the implantation in a sequential lecture. I'll upload the implantation proper.